Dr. Douglas Axe is a molecular biologist and chemical engineer, a Caltech PhD, and was a postdoctoral research scientist at the University of Cambridge in England. He is the director of the Biologic Institute in Redmond, Washington.
What I'd like to talk about today is a question that comes up frequently for people who work on intelligent design, and that is people are very aware that part of what intelligent design people do is demolish Darwinism, but they're not sure what the positive agenda is. And that comes up with the question of God of the gaps as well, I think. That if the perception is that design is simply what you have left over when you've nuked Darwinism, then it doesn't have the feel of a positive theory in its own right. And it does deserve attention as a theory in its own right. And the people who are approaching this, people like William Dembski, who have framed formalism for detecting design, are thinking not really of Darwinism, they're thinking of how do you do design. But it is true, I would say as a biologist, that we're at the infancy of developing a new way of doing biology. It's not there yet. So in a sense, you're right when you say where is the new construct that would replace Darwinism? It's not there yet, but we're working on it. And I want to talk about hints of this new construct today. But here I just want to show with these, there's a building being torn down and a building being constructed. And if we assume that the new building is going up on the site of the old building, then you would take care how you demolish the old, because you could drop a nuke on it, but you have this huge crater you have to deal with and kind of a death zone around it, so it's going to be hard to get people in there to work on the new building. So I think there's an analogy here with theories that it's one thing to deconstruct and demolish a theory, and I am interested in doing that, but I'm not interested in doing it just for the sake of beating up a theory. I and others are wanting to prepare the ground. Demolition is part of what you do if you want to build something new. Another interesting aspect of this is if this building is being torn down because something went wrong structurally, it could no longer be used as a building because it's unsafe, it's falling apart. One of the things you might want to do is, as you're taking it apart, learn about what went wrong with the old building, and that might factor into the design of the new building. So that serves as an analogy for what I'm going to take forward here. And what I'm going to do is very quickly, because we don't have much time, touch on a number of key problems that have been identified with the Darwinian paradigm. All of these have led to publications, technical publications. I'm only going to touch on them briefly. But after I walk through these, there's 7 points where we found significant failings of the current Darwinian paradigm. I want to then go back through the 7 and talk about how you can reframe the failing in terms of a positive design principle. And these are really simple, and I'm not going to call this a theory yet, but it gives you an idea of how you can deconstruct one theory and use the failings of the old theory to help you think about what the new theory should look like. Okay, so the first critique from this is that new gene functions must offset their cost. There is a cost to carrying a gene, and that cost must be offset from the very beginning. So any evolutionary story that talks about new genes, it has to talk about from the very beginning. As soon as they are functional, they need to be functional to an extent that offsets the cost. Okay, these are the molecules that I referred to yesterday. 2 very similar proteins that perform different chemical functions in the cell. And we did a series of experiments where we simply asked, can you convert this by mutation to perform the function of this? And because they're so similar in structure and in sequence, you would think that the answer is yes. And clearly it is yes. If you did enough changes to the one on the left, it would perform the function of the one on the right. We were asking, can it be done in few enough changes that this thing could happen on an evolutionary timescale, even on billions of years? And we found that it couldn't. The paper describing this result, even after we changed the active site, the inner cleft that actually does the chemistry, changed all of the amino acids in the active site from A to B to make it resemble the target enzyme. It still didn't perform the target function. So we inferred from this work that it would require more than seven or more specific changes to the gene in order to convert the one into the other. If you do the math, again, 7 doesn't sound that remarkable, but remember the example from SMU, 11 changes to the sentence is exactly the same problem. Once you have to make 7 specific changes in a gene, it requires a tremendous amount of time for that to happen, and more than there is time in the history of the planet. Second critique, converting existing proteins to new functions typically requires extensive reworking. It's not a simple thing of just changing 1 or 2 amino acids. Another problem, which I refer to as boom or bust, or feast or famine. First of all, let me say that if you're going to get new genes to evolve, it will happen in bacterial populations. Everything else, more complex life, it becomes much, much, much harder because bacteria are far more abundant. They have a much shorter generation time. You just have far more possibilities to evolve things in bacteria than in complex life. But here's an aspect of the way bacteria propagate and maintain their population on planet Earth. Because they can replicate so quickly in less than an hour, you have 1 bacterium in half an hour, you have 2, half an hour later you have 4 under good growth conditions, which means in a few days, under the right growth conditions, 1 bacterium could repopulate the planet, which is remarkable. In less than a week, 1 bacterium, if you gave it the right growth conditions, could produce a global scale population. There's no other organism that can do that. Complex life can't come close to that. What that means is the way bacterial populations are stabilized is that certain cells are lucky and most cells are unlucky. Some bacteria that are alive today will have billions of descendants alive a month from now, but for every one of those, there's a billion that will have no descendants alive a month from now. And what that means is this. If you want to talk about evolving something new and clever, you're talking about these rare events happening in genes. It turns out because of this feast and famine phenomenon, you could have these rare events happening, and just because they happen in the wrong place, they will be swept away, even if there is a slight adaptive advantage. This is a well-known phenomenon. What it means is you have to have these things happening over and over and over and over and over and over and over again before one of them will happen to be in the right place to have billions of descendants and to sweep the population. Okay, and this is not controversial. This is well recognized. I've described this in a population model that's also been published in Biocomplexity. The third critique then is that innovations, these new inventions, they have to be surprisingly common if they are eventually to become established, to become fixed in a population. And it turns out, if you look at the numbers for bacteria, if you want to say that some remarkable thing evolved within a multi-billion year time frame, what you're really saying is it's out there somewhere almost every day, waiting for that lucky break when it will sweep, okay? So that's an interesting perspective because people are willing to say that all kinds of things could happen in 5 billion years. What we're saying is the math shows they should be out there somewhere right now if they're going to happen in 5 billion years in a bacterial population, which is a very interesting point. Let's move to protein folds very quickly. I've done work on the origin of protein folds, how hard it would be to get an entirely new structure. So remember the purple and the blue, those two things look very similar. They perform different functions. But where do you get something totally radically different that's not like either of those? There are thousands of these folds that have been cataloged. The way I approached this experimentally was to take one of these folds and to shuffle the amino acid side chains within groups. So I've shown 4 colored regions where I did the shuffling, produce a library of these variants, put them in cells, and find out what fraction of these work after you've done the shuffling. If you do that, and you do a little calculation after you get the results, you can calculate how rare functional sequences are in the whole space of possibilities. In other words, you can estimate how hard it would be to randomly pick sequences out of the hat and get one that folds and functions. And the result was published in 2004 in this paper in Journal of Molecular Biology. Here's the end of the abstract. The conclusion is this result implies that the overall prevalence of sequences performing a specific function by any domain-sized fold, it was 150 amino acids, may be as low as 1 in 10 to the 77th power, adding to the body of evidence that functional folds require highly extraordinary sequences. I don't know if 10 to the 77th power means anything to you. It probably doesn't. I sometimes give a visual illustration of what it means to hit a target that amounts to to hit a bullseye. No, a target that amounts to 1 part in 10 to the 77 of the whole possible space. So, any archers here? If you're an archer, you've got the target out there. I'm going to assume that this guy's competent enough that the arrow is going to go forward, okay? And we're going to assume that the target amounts to 1 part in 10 to the 77 of this whole scene in front of him. And I want to show you how hard it would be to hit a target of that size. We're going to put the archer right about out here. Got his oxygen tank, got the arrow ready. And we're going to say, I'm going to tell you what the target is, and you need to hit it in 1 shot, okay? Tell me what the target is. It that galaxy? No, but it's in the galaxy. If we zoom in a little bit, you'll find that there is one particular star. I have to hit that one particular star. Well, hang with me. There's a planet that orbits that star called Earth. I have to hit that planet from here. Hang on. There is a region on the planet, Lake Michigan. I have to hit, no, hang on. Chicago? No, a park in Chicago. Hang on, a little bit better than that. Okay, I have to hit a dude from out here in outer space. Not just that, you have to hit his hand. No, I've got to hit a cell in his hand. No, the DNA in the cell. No, a particular carbon atom in the DNA in a particular cell of the dude's hand. No, the nucleus of the carbon atom of this particular, no. See, the nucleus is protons and neutrons, right? I have to hit a particular proton. No, it's not that easy. You've got to hit a quark in the proton. So I don't know of any arrows that have tips that are that fine, but that is the problem that you're faced if you have to hit a target that's 1 in 10 to the 77th power. It's ludicrously implausible. Fourth critique, new protein folds require completely new designs, and there's just no way from everything we know that chance can accomplish that. Okay, fifth critique, new metabolism requires the invention of multi-step pathways. If you're familiar with Behe's concept of irreducible complexity, that's what we're talking about in metabolism. Let me give you another problem that I've called causal circularity. It's also described in this forthcoming paper. This is a picture of an enzyme that's called sulfite reductase. It's a bacterial enzyme, and it's important. You've never heard of it, but you depend on this enzyme because this is the enzyme that brings sulfur from inorganic sources into organic chemistry. And there's 2 amino acids that all of life depends on that incorporate sulfur. One is cysteine and the other is methionine. 2 of the 20. And this is the guy that gets the job of pulling sulfur from from inorganic sulfur sources and brings it into organic form so that we can, and all other life forms, can have those two amino acids, cysteine and methionine. And cysteine turns out to be it's the smaller of the two. Cysteine turns out to be the entrance of sulfur into life because methionine is made from cysteine, okay? And this is the enzyme that pulls it in and makes cysteine. It's on the cysteine biosynthetic pathway. It has some interesting features. The green parts here are the protein part, but a lot of these proteins, they bind things in their active site that allow them to function. This looks like heme and hemoglobin. It's cyroheme. It's very similar. But you notice there's a little cage thing here that looks like a cube, which is called an iron-sulfur cluster. These two things are absolutely essential for this sulfite reductase to work, which means they're absolutely essential for life because this is where sulfur comes into life. And this little cage here, it's the 4 red corners are iron and the 4 greenish yellow corners are sulfur. It's an iron-sulfur cluster. It has to be coordinated to the protein, and the protein uses particular amino acid side chains to hold this in place. And if that cage is not there, we're all doomed, okay? So my question is, what do you think are the side chains? Which amino acid do you think might be grabbing this and holding it in place to do the job to make cysteine? The answer is cysteine, okay? So think about that. If you could explain somehow how a primitive bacterium could get the gene to make this protein to make cysteine, it wouldn't work unless you also gave it cysteine. It would not work without cysteine. We call this causal circularity. It is not unique to this pathway. This turns out to be a very general phenomenon in life. You need cysteine to make cysteine. You need lysine to make lysine. You need arginine to make arginine. In fact, I've given this a general form. The biosynthesis of X requires X. This turns out to be very general formula in biochemistry. In fact, if you think about it, this is going to be true for all essential components of life. To make DNA, you need DNA. To make RNA, you need RNA. To make proteins, you need proteins. This is true. It's a general pattern of life. This shouldn't disturb us as people who design things, because there's all kinds of things that we design that you need to get the whole thing together to work. But this should be very disturbing to someone who thinks that life came about through a material process. Sixth critique, life depends on an extensive network like this of interdependent functions. It's completely plausible and familiar from a design point of view that you have this integrated thing, the whole thing has to be working together. That's familiar to design, but there's no way you can cobble it together by accident. Last thing I'll talk about, and then I quickly need to run through the design principles, is at the biosphere level. And this is a paper came out of this that I wrote with Matti Laisala, who's in Finland. He's an expert on a molecule that's found in wood that's called lignin. I'm not, but we talked together about this interesting phenomenon that lignin, although it's a hugely abundant molecule on Earth, about 1/3 of the non-fossil organic carbon on planet Earth is lignin. And lignin is the glue in wood and woody plants that holds cellulose together into this matrix that makes the woody stems, not just in trees, but in bushes, any large plants. They depend on lignin for their structure. And the interesting thing about lignin, it turns out to be hugely important for complex life on planet Earth, because lignin, when plant tissues decay, stems, twigs, trees decay, lignin is broken down, but it's broken down very slowly, and it's crucial for forming this top humus layer in soil where you get air and the right composition of soil in order for plants to grow. And plants, I don't know if you've noticed, but plants are pretty important. All of complex life depends on plants, which depends on soil, and depends on soil having this layer, this organic layer on the top, which depends on lignin. Specifically, it depends on lignin being retained in the soil and slowly breaking down. Now, here's the interesting paradox that we posed. If there's so much energy sitting there in lignin, why hasn't some greedy little microbe evolved the ability to eat lignin for lunch and have a heyday? 30% of the non-fossil organic carbon on the planet is in the form of high energy lignin. It's higher energy than cellulose. The only more abundant organic polymer on the planet is cellulose, but lignin's even higher energy. If something evolved the ability to chow lignin and derive energy from it, it would have a ton, I'm not exaggerating, billions of tons of lignin to dine on, and yet nothing has. If it did, if something rapidly degraded lignin, you would not get this humus layer in the soil, and it would be the end of life as we know it, Jim. You would not have large plants, you would not have complex animals. It would be a disaster for the planet. Nothing has done this in the 400 million years that lignin has been present. So, seventh critique, a mechanism that leaves low-hanging fruit, stuff that should easily evolve, unpicked, can hardly explain how so much high fruit has been picked. There's a contradiction here. If you want to say Darwinism is a mechanism that can accomplish anything, and it would seem that it can if you look at everything that it has accomplished, how do you say that it didn't accomplish these things that it should have accomplished? And it's interesting that the things that it didn't accomplish are exactly the things that would be a disaster, and the things that it did accomplish are the good things. That doesn't fit very well with a materialist theory. So let me now quickly shift and go into construction mode. And all I'm going to do is run through these critiques and give you a one-sentence principle that we think hints at a design principle. First critique is that new gene functions must offset their cost from the very beginning. That's familiar. The principle we use here is inventions usually require rounds of testing and refinement before they're ready for launch. This is how, you get investors when you have an idea, and the investors are willing to put money into the idea before it pays off. That's familiar to the design world. Second critique, converting existing proteins to new functions typically requires extensive reworking, and the principle is that design concepts are readily adapted to work in different contexts, but material things aren't, okay? Here are two material things, surgical scissors and tin snips. They work by very much the same physical principle, but if you were being wheeled in to the operating room and you saw the tin snips on that tray, pull the things out and get out, okay? Same concept, but definitely cannot use one to do the job of the other. Third critique, innovations have to be surprisingly common, always out there somewhere if they are eventually to become established. The principle is that even good innovations don't sell themselves until they reach a critical level of exposure. And if you know technology and entrepreneurship, there are a lot of good ideas that they're good ideas, but they don't make it unless they get into the right hands and get in the right places. That's a familiar idea to us as inventors. Fourth critique, new protein folds require completely new designs, which can't happen by chance. The principle is that radical innovation requires completely new ideas. It's not just a morphing of the last idea. It's insight. The telephone. There was nothing like it before. Alexander Graham Bell came up with the telephone. Radical insight. Fifth critique, new metabolism requires the invention of multi-step pathways. And the principle that goes with it, a design principle, is that new technology requires the solution of many problems, not just one. And this is very familiar. So if you think, going back to the telephone, the jump from landline telephones to cellular, it involves all kinds of innovation in the handset, but not just in the handset, in these the whole system where you're receiving these signals, the towers, the whole thing requires innovation. Sixth critique, life depends on an extensive network of interdependent functions. This is the causal circularity thing. The principle, system design is approached from top down with the function of the whole in mind. So, sticking with the phone, the jump from cellular to smartphone. None of these happened by a simple tweaking of a cell phone, or it would have happened earlier. It took thinking outside the box, top down. The iPhone came out and that changed everything for smartphones. Seventh critique, a natural mechanism that leaves low-hanging fruit unpicked can't explain how so much high fruit has been picked. And the principle is that good design is as much about what is not done as what is done. And so, sticking with the telephone, you can imagine all kinds of combinations of functions that could be put together, but just because two functions could be put together doesn't mean that they necessarily should be put together, okay? Some of these would be silly to put them together. Okay, so with that, I claim that we have now examined on a number of different levels whether the Darwinian paradigm works, and it has failed on all of these levels. But I also think we're seeing a tantalizing beginning to the weaving together of ideas that are not simply destructive, not just taking down the old building, but asking what was wrong with the old building and weaving together something that looks like a new building. And it's not there yet, but it's on its way. And so this is the exciting thing that we're moving into. And with that, I will end and take any questions.
To view this program again and share it with your friends, and to learn more about this series, these conferences, and the intersection of science and faith, please go to www.pensmore.org.